feel free to start your presentation and discuss your work. You can also introduce yourself, so then we know.
Sure. Thank you. Thanks very much for Professor Testas for your invitation. I'm really my great pleasure to present our work. So I'll just start to share my screen. So is there, right? The screen is.
Yes.
Cool.
No,
so
wait, not yet. I would usually take a few seconds, and then it should appear. Yes.
Okay, cool. So I'm Kai Wang from Institute of Neuroscience. Now, a new name is Center for Excellence in Brain Science and Intelligent Technology, Chinese Academy of Sciences. So it's my great pleasure to share our recent work on a new type of light field microscopy to imaging zebrafish and mouse brain. So a little background. And people all know that optical imaging is a very powerful tool to study biology science when it is combined with powerful fluorescent indicators. And applying optical imaging for brain science has even more challenges because brain is a neural network consisting of a huge number of interconnected neurons. And the information processing in this network happens in very small units over large scales and high speed over a long period of time. And even more so, usually brain functions normally and optimum in their natural environment and the behavioral states. And also, if you want to study some higher order cognitive function, you have to use more higher order animal models, etc. So all those very challenging requirements are very hard to be met by a universal optical imaging solution. So modern optical imaging technology are developed to meet certain requirements. For example, so previously I have been working with on the adaptive optical microscope, try to imaging deeper to have a higher resolution deep into the tissue. But today I'm going to focus on developing, talk about the technology to improve the imaging speed, focusing on speed. And one very effective strategy to improve imaging speed is try to increase information parallelization. So we compare several popular imaging technology, including, for example, Confocal and photon, which is based upon scanning and collection information serially. It's relatively slow. For example, light field microscopy is relatively faster because it parallelizes information collection in a plane. But if you want to image in a three-dimensional volume, you still have to scan in the third direction. While the light field microscopy is very different. It's a scanless 3D imaging strategy. It does not have to scan, and it fully parallelizes the information collection. So actually, this idea was originally developed more than 100 years ago in the field of photography. But it was recently reintroduced into microscopy and attract a lot of people attention recently. So implementation of the conventional light field is very simple, as shown in this picture. I hope you can see my mouse. So you just insert a lens-lit array as an imaging plane of a conventional wide field microscopy and move back the camera sensor to the focal plane of this micro lens-lit array. Then you have a light field camera, a microscopy. It just not only collects the information of how the light ray intercepts on the camera, but also collect the information, the direction of this light ray. So they can just take one picture and do the three-dimensional reconstruction, digital refocusing afterwards. But so far, everything is based on a light ray model. To increase the resolution, people have provide a full wave optical description of the light field microscopy introduced by Broxton in 2013, and which achieved the higher resolution. And soon, a lot of people applied this technology to imaging, for example, zebrafish brain and mouse brain. And but there's several problems. One problem is that the resolution achieved, for example, in previous demonstration is not optimum. And second, they have a reconstruction artifacts near the naive focal plane, as shown here in this picture. There's an artifact. So, in order to get around of this issue, our lab several years ago developed a light field camera microscopy called the extended field light field microscopy. Term is XLFM. Actually, we have this idea by accident because when I was working on adaptive optics. So just let's look at in the adaptive optics. There's a very well-known device called Schick-Hartmann wavefront sensor. So if we compare this Schick-Hartmann wavefront sensor, it's very similar to light field microscopy. It also has a lens-lit array, but this lens-lit array is placed as a focal plane, not as an imaging plane. So the way it works in the adaptive optics is that if you have a point source here, then on the camera, you will have a array of points. That's the image formed by this lens-lit array. And depending on how this array changes, you can infer what's the wavefront aberration. So to make this device work, which is originally introduced in astronomy, in that case, you always have point-like source. But when we try to make it work in the biology sample, we worry that whether we can still have a very nice point source for wavefront measurement. So we asked the question that, what if this is not a point source? What if it is a three-dimensional structure? Can we still do a wavefront sensing? Actually, after asking this question, we realized that actually this Schick-Hartmann wavefront sensor can do three-dimensional volume reconstruction, which also realized later by by other group that the term it so-called this Schick-Hartmann wave sensor can be termed as Fourier light field microscopy. So how it works? Just take a look at the raw camera frame. So this is a picture we get on the camera frame, and this is not point source, it's a three-dimensional zebrafish brain. So you see a lot of zebrafish brains. Actually, each small image of this zebrafish brain is the projection view from a certain direction. Then you can use the very similar concepts like in CT, the projection tomography to digitally reconstruct the three-dimensional volume. So you have an object viewed from different direction, and then you can digitally reconstruct the 3D volume. That's the projection tomography. Okay, but this technology gains speed at the price of reduced resolution. So what's the resolution in light field in this light field microscopy? Let's look at the schematics. So imagine that's the objective. We have a object of 3 points in 3 dimensions. So imagine we have 3 microlens placed at back pupil plane. Then we imagine these 3 points at different directions. The image we collect on the camera through 3 different microlens will be like this. So because this is a view from top, so the red dots and the yellow dots overlap from each other over each other. So the lateral resolution actually determines whether we can separate this, for example, the red dots from the green dots. That's easy to tell because that only depends on the corresponding numerical aperture collected by each microlens. And we can have the RB limits equation that the lateral resolution is determined by the numerical aperture of this microlens. Well, what's the axial resolution? So using this macro lens, the central macro lens, we cannot separate the red and yellow dots in the axial direction, but we can resolve them using this macro lens because they view these 3 points in a different angle. And do some simple geometry calculation, we can get that the axial resolution is actually the lateral resolution over tangent theta. Theta is the angle of the light ray would collect by this macro lens. And apparently, this resolution cannot maintain over the whole volume. It only maintained over a so-called Rayleigh range of each beamlet collected by each macro lens. So that's these 3 parameters fully describe the performance of a light field microscopy. Okay, in this method, we just take one picture, we get a volume, and it can be run very fast. For example, we can image fast circulating blood cells in a living zebrafish brain, and which is very challenging for traditional scanning-based imaging technology to achieve. Well, even more interestingly, we try to find an even more interesting application. We are lucky to met Dr. Wang, who is in University of Science and Technology of China. He is working on the 3D moving zebrafish tracking, trying to do real-time behavioral analysis on this small fish. So we met each other in a meeting, and we decided to combine their tracking system with our imaging system so that we can imaging a whole brain neural activity in a 3D moving zebrafish. One interesting phenomenon behavior is that free moving zebrafish can pre-capture paramecium. And this involves a lot of neural information processing, such as the motivation, the model preparation, river learning, etc. And we worked together, and we did make this work. On the left bottom corner, that's a behavior video. You can see a freely moving zebrafish can capture a paramecium, and at the time we do the whole brain functional imaging, we can see a lot of brain regions light up during this process, which is not possible if you just immobilize zebrafish larvae in agarose or etc. Because in that case, the zebrafish cannot move and catch this paramecium. So we can quantify these parameters and align them with calcium activities, and to investigate how these neural activities correlate with behaviors. So as you can see that the resolution in this demonstrated case is not perfect. So we want to improve the resolution. How can we improve resolution? Let's go back to our original camera frame as we show that this is the raw camera frame we captured. That's the information or 3-dimensional reconstruction we are starting from. So if we look at the one particular small image, as I explained that, which is actually a projection view of the zebrafish brain from a certain direction. So one micro lens corresponding to one particular direction. And in our previous design, we tried to cover the whole zebrafish brain. So we want to have an optimal resolution coverage over the whole brain, so that the beam in that design, the beam has small numerical aperture. So that the Rayleigh range of that beamlet can be optimized to cover the whole zebrafish brain. In that case, the lateral resolution cannot be very high because we require the Rayleigh range to be large enough to cover the zebrafish brain. Well, if we want to increase resolution, optically is very simple. You just have a different design. You increase the numerical aperture of each macro lens, so that in that way you can increase the resolution. However, the Rayleigh range of this beamlet is reduced. but that's not the worst problem. The worst problem is that the background information away from the focusing volume, because the Zebrafish brain is larger than the axial coverage of your light field camera. Those information called background information will come into your camera and cause noise, not only noise, but cause reconstruction artifacts also. So the key to improve the resolution is trying to get rid of the background. So how can we get the background? Actually, if we look at all modern optical imaging technology, we are all fighting with the background. For example, Confocal uses a mask to get rid of background. Multiphoton uses nonlinear effect. Light sheet uses selective excitation. Actually, people in the light field camera imaging has already tried to get rid of background using different strategies. For example, people have combined temporal focus and multiphoton excitation with light field camera. But in that case, it's very hard to use nonlinear excitation to cover a large volume and to provide enough photons to image at high speed. People also introduce light sheet imaging, light sheet excitation to confine the excitation over a selected axial range. But this configuration need to introduce the light from the side. It's not directly compatible to achieve imaging of freely moving zebrafish or to imaging large samples such as mouse brain. So we want to combine the confocal concept with light field imaging. But how can we combine that? We realize it's not easy. We have to generalize the confocal detection strategy. So just let's look at these schematics again. So imagine that we have a sample. We have a focus volume in yellow, which we want to get information, high resolution information. The red and the cyan that is above and below the focus volume are just background we want to get rid of. So in our conventional light field imaging, we use wide field excitation. We illuminate the whole sample altogether. That will cause background. The background cannot get out. We cannot separate background from the in focus information. But if we choose to use just conventional tightly focused spot, confocal excitation, we realize it also will not work. It still cannot separate information from different depths. So it took us several times. Actually, it's just very simple. It turns out they just use the vertical light sheet, vertical slim light sheet to excite the whole volume. So in this case, then let's look at what happens if you use 3 microlens to image this vertical plane. And then, okay, because you're imaging this vertical plane from different direction, central lens still form imaging like this, but the macro lens on the edge of the objective imaging actually view this vertical light from this direction. So the imaging has a shape like this. So it's the vertical information is spread laterally. And then you realize that if we put a physical mask here, and except for the central one, the focusing information will pass through this mask, and this mask will block the background above and below the focusing volume. So, and then we can scan this vertical light sheet to cover volume. To make this work, we demonstrate that that's a 3-dimensional animation showing that's the mask for example 7 microlens, and that's a vertical light sheet, and then we can sorry. So I hope you can see this video. When we when we scan the light sheet and we also scan this mask together, then we can extract the focusing volume in yellow only and blocks most of the background in red and cyan. We test this idea on a densely beads embedded in 3D agars. And you can see that without physical mask, the image you collect on the camera is very noisy, background dominates everything. You can not get high resolution information, but with mask, you can get rid of this low resolution background information, can preserve this high resolution in focus information. That's the key, and that concept is demonstrated. But then we still have the problem that because when we get the high resolution information, we have reduced the axial coverage. Therefore, we have to scan to have a very fast axial scan strategy so that we can cover a larger extended axial range. So how can we do that? Traditionally, people usually trying to use a piezo state to drive this objective up and down, which, however, is not fast. The speed of light field can be very fast. That piezo speed cannot match the light field imaging speed. Therefore, we try to look for a different strategy. We insert a piece of glass here, and then this glass will shift the focusing volume away from their original position. Then on the camera, we'll have a different position. Sorry. And then we can shift the focusing axial range by just putting a piece of glass. And if we can insert and remove this glass at a high speed, we can scan in Z direction at high speed. So finally, we integrate all the idea together using this is schematic of our setup. So this is objective, and this 4F system, we conjugate the micro lens array to the back pupil of the objective, and we put this is physical mask, and this is a glass plate mounted on a rotating wheel. So when this wheel is rotating, the mask and the glass will rotating at the same time. And we have even more animation for that. So you can see we use a gavel to scan the exciting laser together with the mask. So the mask is on a rotating wheel, and we have a different glass plates of different thickness. So in this case, we can scan in axial direction at very high speed. So have all this problem solved. We image the Zebrafish again. We try to compare with our previous version of Light Field. This previous version has relatively low resolution. Those are all the same Zebrafish line with cytosol GCAM labeling. So this is a higher optical resolution, but without physical mask to reject background. And we see that only if we not reject the background, we can get this high resolution and improve the signal to noise ratio. Okay. So we also apply the technology to image the free moving zebrafish during its pre-capture behavior. You see 5 different images that's corresponding to 5 different axial scanning position, corresponding to 5 different glass plates. Okay, so at the end of this movie, it's a relatively long movie. So at the end of the movie, you can see that zebrafish successfully captured paramecium. So, okay, let's play it faster. Okay. Oh, okay. Trust me, it. And then we can analyze data, and it has much improved resolution. as evident from, so this is zebrafish whole brain before and after successful prey. And at different axial range, we can see many isolated bright spots indicating isolated single neurons that is active during this process. We see a lot of features. If we zoom in, for example, we can see small regions in the upper tectum corresponding to the visual response. We can see single neurons activation along in this process, and etc. Okay, so in this experiment, demonstrate that confocal light field can provide improved resolution and signal-to-noise ratio in imaging free moving zebrafish. Then we want to focus apply this technology to imaging mouse brain. But in mouse brain, previous system is a little bit complex. So that is because we have to do fast axial scanning. If we do not need fast axial scanning, the simple system can be greatly simplified. So in this system, we have we do not have axial scanning, so that in this case we have a mask that is stationary. It do not have to be stay on a fast rotating wheel. We use two gavels. One gavel is for scanning the excitation beam and to cover volume. So also the fluorescence is descanned by the same gavel so that the mask can stay here stationary. And then we use a second gavel to rescan the image onto the camera. And using this technology, we can record calcium activity over volumes in the mouse cortex. The speed actually is limited by camera, but the calcium is relatively slow. So we can run, we usually, we actually, in the end, we decide to run at 5 to 10 Hertz to match the speed of the calcium. And we try to compare the image captured by the confocal light field with the golden standard captured, for example, 2-photon microscope. We see that the neurons found in these two imaging modalities are very close to each other. And if we check the whole volume in 3-dimensional, so that's virus injection labeling strategy. And we see a lot of labeling into layer 2, 3 in the mouse cortex. Layer 4 is not labeled well. And label 5 has several sparse labeling. And if we compare the imaging volume captured by the confocal light field, we can see that most of the layer 2, 3 neurons can be well recorded in the confocal light field. The maximum depth we can achieve is about 370 micron, which covers the layer 2, 3 neurons in mouse cortex. Also, we apply the technology in the mouse can do imaging fast circulating blood cells. So for example, so each volume, because it's confocal, it has a limited axial coverage of only 150 micron. So we image at different depths. As we image deeper and deeper, the background becomes an issue, and confocal can allow us to remove the background so that we can image up to 500 micron deep into the mouse cortex. Another reason that we can image such deep is that we use a near infrared dye to label the blood cell, so that we can pretty much track, because of the high speed of the light field, we can pretty much track every single neurons in the capillary, not in very large blood vessels, but in these small blood vessels. We can quantify the moving speed of this circulating blood cell. For example, if we focus on one particular blood vessel and we plot the intensity along its distance, we can see this very typical trajectory of the blood cells. We can quantify their velocities. We can see their velocity is changing. Because we get a 3D-dimensional volume, we can also quantify how the blood cells is distributed across these branches. And we can also quantify their average speed over time, over population, etc. So that's pretty much the work today I'm going to introduce you. So to summarize, actually, I introduced two work. One is our previous published XLFM, which we demonstrated the whole brain functional imaging in freely moving lava zebrafish. And then we improve the resolution and signal-to-noise ratio by introducing a generalized confocal detection strategy. And we demonstrate improve the resolution in free moving zebrafish. We also demonstrate the imaging calcium and the circulating blood cells in living mouse brains. So those work are primarily done by research assistant Lin Cong in our lab, and the very talented students Lu Bai and Jinkun Zhang, PhD student in my lab. I would like to thank you very much for my collaborator Quan Wen from USTC, and also Misha Aaron for the zebrafish, and Ed Boyden for later light field imaging collaboration. Thank you.
Thank you, Kai. Thank you for the wonderful talk. It was really, really exciting method. I mean, to see how microscopy is moving more and more to be able to speed, high speed, and be able to measure like a freely behaving animal, and that was really, really great. So I'm gonna read soon the questions that are being posted in the live chat, but maybe I first start with one question myself that could be a little bit biased by my background in the field of super resolution microscopy. So, but we know that like in our field, we deal at the end with very few molecules, so with very few photon detected, right? So how much is your signal loss, fluorescence loss in the detection path, for example?
So, right. So we actually, let me see whether I have slides here. So for example, if you look at this, we use in order to get high resolution in the axial direction, because the axial direction is determined by the angle. Largest angle we can collect on the edge of the micro lens at the edge of the pupil. So we usually use a large numerical aperture objective as large as we can. And then their strategy, whether you put the micro lens, densely packed micro lens, or a loosely packed micro lens. We decide to use a loosely packed micro lens because we do not want too much overlap between sub images. And but we did the calculation, for example, how much light is collected in our setup. Equivalently, although we use, for example, 1.1 NA objective, the light collection efficiency of our system is equivalent to objective of 0.4 also, depending on different design. We have a 0.4 version, we have a, for several different design, we have a 0.5 version. It depends. So, right. So we didn't optimize desperately on photon collection. But for single neuron resolution, calcium active detection, so the current design is okay, it's enough. So okay. I hope I have answered your question.
Yes, yes, it did. And then maybe another curiosity, like to get axial resolution, you had this nice idea of using the different glass rotating. Okay. How does that compare, for example, with the use of tunable lenses?
Right. So that's right. So we also think about using tunable lens. But we did the calculation. The tunable lens, the tunable range is not enough for a high NA objective to cover several micron axial movement. So for low NA objective, the axial shift of several, for example, 200 micron, you can use a tunable lens. That tunable range is enough. But for high NA, for example, like 1.1, the tunable lens is barely enough. And also the aberration it might cause probably will we might need different mask. So if you have an aberration, those spots will shift their position, and you might need a different mask to get the right to extract the right axial range information from a right axial range. So that's also consideration. We decided not to tunable lens. We just use glass plates. So indeed, when we use different glass plates, we actually have each glass plate. We have a dedicated mask for this glass plate, so that the match between the mask and the glass plate is perfect.
I see. Well, thank you. Okay, now I'm going to move to reading the question from the live chat.
Sure.
I'm preparing you that we usually get a lot of questions, so I know you're already sleep deprived, but I hope you can resist a few more minutes.
I'm excited. I'm not that sleepy at all.
Okay, that's good. So, so the first question is from David Shopik, and he's asking, could a special light modulator, an XLM, work to mask the light field image instead of the rotating wheel? So why not?
Special light modulator to work as a mask.
Yeah.
Then I would think he might talking about amplitude special light modulator.
Maybe.
Right. If you have so the mask, I think yes. If you have a flexible, tunable, like special light modulator, you can to draw apertures at will, all different types of apertures at will. I think it will add a flexibility to the system, but you still have to shift the focus. I think shifting focus cannot be easily achieved by the special light modulator. The glass plate here is functioning by shifting the focus. Right. So I don't know whether I answered the question correctly, but right. I think special light modulator itself may not be able to shifting the focus. You have to use some other way to shift the focus, and then you can use the flexible tunable mask to block the light at your will.
So we have then another question from Taras, which who say great talk. Also
Thank
really loved your slide design. So he's asking if are all lenses in the array of the same numerical aperture? And if so, what would happen in case lenses have different numerical aperture?
Good question. Right. A very good question. So yes, we have the exactly same numerical aperture in all our lens design. So I don't have. Let me see. Okay.
So, he's also asking on the way if the fact that have different NA can improve in some way the resolution also.
Right. So we do have we introduce diversity in terms of different microlens into our system in our original design. So the diversity we introduce is try to have two different focal lengths. The reason for doing that is that we want to have two different group of lengths. One group of lengths focusing at a certain axial range, and another group of lengths focusing at another range, so that each of them have a relatively high resolution, and then we can tie them together to have an extended focusing range. That's the one diversity which we introduced in our XLFM. When you're talking about have two different NA, then the high NA group will achieve higher resolution over a reduced axial range. I think you need a very special application where, for example, you want to get a global picture over a large field view at low resolution, but at the same time you want to get the very high resolution over smaller volume at the same time. Then you can design your light field using that strategy to use two different group of lengths. Use low numerical aperture lens to cover larger field view, but at low resolution, high resolution, high NA lens to cover reduce the volume at a high resolution. And the computation algorithm, you can very easily to integrate these two groups of information coherently and to achieve optimal performance. That's no problem in terms of reconstruction algorithm. You can collect all the physical, all types of physical information as you want, and you can combine them together coherently using algorithm. It's just what really you want to get. You can design your system at your will. That's up to you.
Yeah. So then we have a question from Aureliana, who is asking, can you measure aberrations from the position of the images created by the lenslet array?
Oh, that's a very good question. Right. Actually, at the very beginning, when we working with Shikam Wavefront sensor, we'll ask the question whether we can use a three-dimensional object to do wavefront shaping. That's the question we asked very originally. The answer is that we didn't dig very deep into that direction. The reason is that in Zebrafish, in our previous adaptive optical microscope paper, we show that the aberration caused the Zebrafish itself. It decreased resolution from like 250 nanometer to like 1.5 micron. So the resolution degrade from 0.3 micron to 1 micron or 1.5 micron. But in the light field imaging, the resolution in our original design is like 3 micron. So in this system, the resolution is very low. It does not care. The aberration will not cause trouble to our system. But in terms of just talking about the computational algorithm, whether you can do optimization, reconstruction, and the wavefront detection at the same time, I think original paper by Mark Laville have talked about this issue. And we also think about that we need an iterative reconstruction method, try to optimize a certain metric function. So if there's aberration, the reconstruction will not perfect because the information cannot combine coherently. If we have defined a metric function so that we can optimize those imaging to make them combine coherently, then you can measure the aberration and at the same time you can get a better reconstruction. That's the idea.
Yeah. Great. So we have now a question from Betul, and she's asking, how does fluorescence saturation and animal tolerance of laser power affect the imaging throughput of confocal LFM?
Sorry, so can you repeat your question? How the laser power affect the?
Yeah, so basically, yes, it's like fluorescence saturation and the animal tolerance of laser power. So how does it affect the imaging throughput of LFM? So how long can you image, or how big the field of view?
Right. So one advantage of light field imaging is that it's highly light efficient. It excites the whole volume, and all the fluorescence is collected for imaging. It does not weight fluorescence. So in our design, we use very low laser exposure, and we still get enough fluorescence. So in our, for example, design, in our first paper to imaging zebrafish brain, I can't remember, but we imaged its mouth 70% also. So it's very light efficient. It has another so because we captured the pre-capture behavior of zebrafish, which means fish not only tolerate this laser exposure, but it can also behave normally to do pre-capture. So that's another fact to show that light field is not very invasive.
So then we have Christina, who is asking if can the resolution be changed in LFM by modulation of parameters such as cellular membrane fluidity, cellular interaction, pH, or electrical charge?
You mean the spatial resolution of the light field? I think in the light field imaging, the spatial resolution is by the optics, the physics of the our microscope, and I think the biology sample might not play a role in this case, but definitely if you can apply several other strategies such as like STORM, you have a low optical resolution, but you can reconstruct a high resolution imaging. But in that case, you sacrifice speed. Then there's no reason to use light field, which is developed for high speed. So I think the answer is that the resolution is limited by the physics of the optical system.
Yes. Then we have Eche, who is interested in so he said that he read that having different openings for different microlens adds light to a point.
Have different openings for different. Can you repeat the question again?
Yeah, so in your work, I read that having different openings for different microlenses adds light to a point. This reduces the background noise, but reduces the depth of the displayed sound. What limitation does this impose on the study?
So, as shown in this picture, we have different apertures for different microlens, and we allow the passenger of light from the in-focusing information. And sorry, I didn't get fully understand the question. So what's the limitation of?
Yeah, basically I think it's like how the having, I think different openings of the microlens. So probably I guess it meant the pitch, but how that impose any limitation on the study. I think probably the opening of the diameter of the micro lenses.
Okay, so let's go back to, so right. Okay. Sorry. I try to find that slides. So I think I get your question. So the opening is choosing to right fit the real range of the detection microlens. So you can make the opening smaller so that you have a reduced focusing volume. But that is not optimum because the light field is designed to achieve high-speed imaging, so it wants to collect a thicker focusing volume as possible, which is limited by the really range of the microlens beamlet. Let me. I don't have a figure. I think I have figures for that.
Oh.
So, as shown in this picture, we designed this size to match exactly the really range of our beamlet. So the high resolution information is within this small range, and we designed a mask to match this small range. If you open the aperture larger, then you allow the background comes in, and then will cause noise and the reconstruction artifacts, etc. So the mask is designed to match the optimal ready range, which is determined by the numerical aperture of each micro lens.
Yes, and then we have Hussein, who is more asking about the application and ask if the method can be used in the detection of neuronal activity in the brain stem and neuronal linkage.
Brain stem, can this technology too? I'm not sure. I don't know. I have no experience to do surgery on brainstem to open the optical window. If you can open an optical window there, then you can use this technology to imaging neural activity there. But compared to 2-photon imaging, light field is 1-photon imaging. It has limited penetration depth of above the in the cortex is about 370 micron. Okay. I don't know in brainstem the tissue will be similar or different. Right. It's to emphasize that light field is 1-photon imaging. The penetration depth is not as good as 2-photon imaging. Yeah. The advantage of light field is trying to go faster.
Yeah.
Okay.
So then Andreas is asking, could you please comment on the volume reconstruction algorithm? The inverse problem is still fairly undetermined. Undetermined, right? How robust is the reconstruction?
Okay, so, right, the inverse problem in this case, if the sample is not scattering, the inverse problem is not difficult. It's similar to CT, but we have a wave optics description, which we use the framework of so-called Richardson-Lucy deconvolution. Actually, in the noise-free, scattering-free environment, it's very robust. It arrives at the very good solution, which is only limited by the resolution, optical resolution of the system. But in scattering tissue, such as mouse brain, it does have a compromised performance. But when imaging mice brain, people have tried to modeling the effect of scattering. Right, I think that is one way to go. If you have a, I think in the scattering environment, the resolution will be compromised. Reconstruction quality will be also reduced. But in a scattering-free, noise-free environment, the reconstruction is robust. It's the traditional rich-lucid deconvolution that has been applied in a lot of microscopy field.
Okay, then Kansu is asking, how do we make sure that the physical mask does not restrict the passage of focused information as well as the background? How do we match the gaps of the mask to the focused area?
Okay, that's if I understand correctly, that's a practical question. As it's a practical implementation. How do you really have that mask designed so perfect to match exactly the lens lead array you have? Right. So the way we build this system is that we build system half with the objective, with the, for example, the objective here, the lens here, micro lens, all in place. And then we put a fluorescent bead to measure a so-called point spread function. And that bead, because that is in real imaging, we also, that's exactly mimic what happens in real imaging. And we put the camera. So in a complete system, it is a mask here, right? But in the alignment mode, we put the camera here, directly measure what those beads looks like on the camera. And then that information gives you very precisely all the information that is all the, so it really tells you what happens in when you're imaging real samples, where you should put the mask. So because we put the camera here to measure what it should look like.
Right, and then we have Andrea question. He's asking, in case of polarized excitation, do the images of the lenslet contain information about the polarization state of fluorescence? Or do you think it could be solved?
Okay, so we didn't take this into consideration because usually in the room temperature, the polarizing information is lost. And I'm not very familiar with this particular field.
Yeah.
Right. So usually you have very low temperature or something, so that the rotation of the molecule is very slow. Then you can measure the polarization. I'm not very sure about this field. So in our case, we didn't consider the polarization effect.
So the excitation is circularly polarized, or
in our case, we didn't pay attention to the polarization of exciting either.
Okay.
Yeah.
Okay. So then we have Iram that is saying, thank you for your great presentation. I'm curious about the limitation of your study and the parts that need improvement in the method.
So limitation of this technology, right?
Yeah.
Right. So as I mentioned that the light field is trying to go for very high speed. So it has, compared to other methods, it has relatively low resolution. If you want to have as high resolution as confocal, then you will not have spin gain at all. You have to sacrifice the resolution to to get it so that you can get the information in an extended axial range. Right. That's a very important aspect. Other things I think including its one photo imaging, so it has the benefit of high speed, but it also has weakness of low penetration depths, etc. Right. So this is a very, very special technique that is only go for high speed. Only in terms of other parameter is not very good. And I think limitations. I think that's my answer to the limitation of this technology. And the second half, I forgot. Is there any second half of the question?
Yeah, it was like the limitation of what would you improve. So what's going to be your next step?
Oh, I think I really want to, for example, we really want to make this technology applied. So our next move is trying to find application of this technology. So for example, to imaging free moving zebrafish, social behavior of zebrafish, etc., and to imaging blood and circulating blood neuromuscular coupling, possibly to simultaneously image the blood cells and neurons, etc. So technology-wise, I don't have a very clear idea what's the next big step.
Yeah, but that's very exciting. So we are looking forward for all this great application, I would say.
Thanks.
Yeah. So that was, I guess, the last question. So I thank you very much, Kai, once again for giving this amazing talk, and looking forward to discuss next time.
Okay, thank you. Thanks very much for your invitation, and it's really my great pleasure to communicate with all you guys. Thank you.
Bye-bye.
Bye-bye.